Hello, hello and welcome back to another video. So in this video, we'll be seeing about chromatography important question and answers. These are basically for interview, whether you are fresher or whether you are experienced and looking for a job into pharmaceutical R&D, pharmaceutical department, for like manufacturing or into analytical department. So this is going to be a very important video. So we'll be seeing into question and answers related to chromatography. So let us get started. What is chromatography? First of all, the question they will ask into analytical R&D is this. Chromatography is a laboratory technique used to separate, identify, and analyze the components of a mixture based on their different rates of movement between a mobile phase and stationary phase. So, right, I have listed down this the definition in very easy way. Chromatography is a laboratory technique, right, where we can separate, identify, and analyze the components of mixture. Suppose there is a mixture of components. Suppose you are analyzing ibuprofen, so there will be different impurities into it, right? Different compounds, different excipients into that compound. So the ability or the technique which is used to separate, identify, and analyzing those components based on different rates of movement means the polar compounds or the non-polar compounds, whichever are eluting first, whether you are using normal phase or reverse phase chromatography, it depends. So the based on their rates, based on the polarity of movement between the mobile phase and stationary phase. So this is known as chromatography. In simple words, chromatography separates the different substances present in a mixture so that they can be identified and measured individually. So I hope this is simple, right? Where we can separate and identify and measure that individually the compounds whichever we are separating. Like example, in pharmaceutical analysis, chromatography is used to determine the purity of drugs and detect impurities. Purity of drugs can be like assay, determining the potency of that drug, or detecting impurities can be related substance test, right? Purity test. What is the principle of chromatography? So the next question they will be asking is this: separation of mixture of components based on equilibrium distribution of analyte between two immiscible phases. So like stationary phase and mobile phase, those are not miscible with each other, right? Stationary phase and mobile phase, and the sample is also not miscible with the stationary phase. So between two immiscible phases, the separation of mixture of compounds based on equilibrium distribution. The distribution equilibrium should be maintained in such a way so that it gets separated easily. So this is very basic, the principle of chromatography. Whether be it HPLC or whether be it GC, stationary phase in the sense we are using column, and mobile phase in the sense we are using different mobile phases with different non-polar mobile phases. So HPLC stationary phase is solid, mobile phase is liquid, as I said, and this is the example of HPLC. GC, liquid is the stationary phase, gas is the mobile phase. This is the example of, this is the photo of gas chromatography. What are reverse phases and normal phases chromatography? These are different types of technique, right? Reverse phase chromatography. So I have listed in such a way so that it will be very easy for you to understand. I have not mentioned it in very complex way so that it will be very difficult. You will not remember and you will not hard to understand those things, right? So reverse phase chromatography, stationary phase is non-polar, like hydrophobic. Hydrophobic in the sense they are water hating stationary phases. They do not dissolve into the water, or they do not stick with the water. Is called hydrophobic, right? So non-polar like C18, C8, C4, different types of columns are there with different chemistries. Might be in other videos I will define about what are the chemistry of columns. Mobile phases like polar, water, ACN, methanol, buffer solutions, different buffer solutions like potassium dihydrogen phosphate or sodium hydroxide or TFA, OPA, different kinds of buffer solutions could be, right? Whether be it sodium, whether be it potassium, whether be it any kind of calcium, different buffer solutions. So what happens is in reverse phase, the stationary phase is non-polar and mobile phase is polar. So like attracts like, we know that, right? So there is a difference in the polarity of a stationary phase and mobile phase, so it will get eluted out faster. It doesn't sticks with the stationary phase present in the column. So that's why we are using a different different polarity between a stationary phase and mobile phase. And our sample, what we have dissolved, is also a polar. Means the polarity index is more difference between each between the solutes. So therefore, the sample also does not get sticks to the stationary phase inside the column, and it gets eluted fast. So if the sample is polar, it gets eluted fast. If the sample is non-polar, in some cases it is also non-polar, or some ingredients, some impurities, some excipients are non-polar in characteristics, so they gets eluted afterwards, because non-polar and non-polar get binds to each other, and it gets and it elutes after, it elutes at later stage, right? In short, I have described here about the reverse phase chromatography and the chemistry and the how it gets eluted the compounds into the system. Normal phase chromatography, stationary phase is silica, polar we know that. Mobile phase is non-polar like hexane, heptane, cyclohexane, cyclobutane, the mixture of hexane and cyclohexane 90:10, 60:40. So that the and our sample is also non-polar, right? So if the stationary phase is silica and the mobile phase is non-polar or sample is non-polar, it gets eluted faster. We'll see about non-polar phases in the upcoming slides. Now, what is chiral or chiral or chiral, whatever, chiral normal phase chromatography, like you seen just now here, right? Last one. So the chiral chromatography is a specialized chromatographic technique used to separate enantiomers. So what is meant by enantiomers? Mirror image forms of the same molecule. Means if you keep a molecule and if you keep a mirror on the other side, how it will look like, right? So the mirror image forms of the same molecule, like ibuprofen or esomeprazole. There are a few examples which are mirror images of each other, right? Mirror images forms of each other, and that have identical physical and chemical properties but differ in their biological activity. Their biological activity differs between each other, but the chemical and physical properties are same. We need to understand this basic difference. Most of the interviews get confused between the chiral chromatography and normal phase, and they get confused what is chiral chromatography. So we need to understand in much better way what is chiral chromatography and why it's important, right? Many drugs exist in two enantiomers. One enantiomer may provide a desired therapeutic effect and other will not provide, right? Other will be less active, inactive, or even cause adverse effects. So we need to identify those, we need to separate those, and that will be helpful for our analysis, which to consider for further drug manufacturing and which to not consider for drug manufacturing. Chiral chromatography helps identify, separate, and to quantify these enantiomers. As I said, how does it work? A chiral stationary phase is used in the column. The two enantiomers interact differently with the chiral stationary phase, causing them to elute at different times. Means the based on the polarity, based on their stationary phase, it elutes at different types. So examples of chiral stationary phase are cellular based column, amylose based columns, cyclodextrin based columns. There are different columns. As you'll come across the information, you'll get to more about this. Hello, so welcome back. We also do support to the pharmaceutical graduates and pharmaceutical professionals. We do have services, right? And we also support students and professionals in 1:1 career counseling sessions and 1:1 interview mock test. So in the counseling sessions, you can learn about your educational qualification. We can talk, right? Learn about pharmaceutical industries, in and out of the pharmaceutical industry, how does pharmaceutical industry operates, what are its benefits, what are its demerits, and how does is the work pressure and work culture, and get to know right career path for you, salary structure according to departments, salary hike tips and guidance. We'll deal and discuss about these all topics and few other topics in the counseling session. We also have mock interview test where we'll do live mock interview test for the candidates who are looking for a job interview and are not able to crack job interview due to some issues, due to some failure in the interviews while at the HR level or at the technical level or the second round phase, right? So we'll get them prepared into 1:1 interview mock test, right? That's so we'll discuss about how to crack in easy manner real-time scenario question and answers, whichever department you are working and whichever department you are looking for, about industry and relative department, about HR question and answer, and how to deal with salary negotiations. So these are the different topics, and few other topics are also there which we can discuss whichever sessions you are taking, and the link is available in the bio of my channel, so you can go and check out that. Is chiral chromatography and normal phase chromatography are same or not? So as we have seen before also, chiral and normal phase chromatography are different, they are not same, because students get confused between these both, whether those are same or not. And in the industry also they use with the similar word, do a chiral test or do normal phase chromatography. So we need to understand the basic difference between these. So chiral means in this we determine the content of chiral isomers, whether is it in normal phase or in reverse phase. The chiral isomers we have seen before, so we determine the content of that into the chiral chromatography. Then only we can say a particular test is chiral chromatography. Normal phase chromatography, in this as said, we must use polar stationary phase, we said, right, and non-polar mobile phase. Non-polar in the sense hexane, butane, butyl, hexane. In that sense we can use non-polar mobile phase. So whether it is to determine content of isomer or purity of analyte, or whatever it is, for whatever purpose, determining content of isomers like the presence of N-butyl hexanes, or presence of any other isomers, enantiomers like what is a phenobarbital or esomeprazole or rabeprazole content, right? So that is called as normal phase chromatography. What is difference between chromatographic purity and relative substances by HPLC? So there in the, if you go in the pharma industry, there they will use a similar term, like you do chromatographic purity or we have to do related substances by HPLC, but there is a slight difference between that, very basic difference which most of the candidates do not know. So you can just read out here and understand. Chromatography is used when all the impurities are known in the given sample. Hence we quantitate only known impurities. Suppose there are 10 impurities in a chromatogram, and if all the 10 impurities are known to us, means those are identified and already we do have COA of those impurities, then those impurities, then that test is known as chromatographic purity. Whereas RS test, related substances test, is used to quantitate both known as well as unknown impurities in a given sample, right? So this is very basic difference. In the RS test we quantitate both, whether be it known or known, but in the chromatographic purity we do not quantitate the unknown impurity. We just catch that impurity and we will just mention that yes, it is separated from a known impurity and it is an unknown impurity. In that way only we will know. So that is known as chromatographic purity. What is a buffer solution? Can you explain about acidic buffer and basic buffer? Buffer solutions which can resist change in its pH value and dilution, or an addition of a solution or an acid or base. Whole pH remains constant is known as buffer solution. Suppose if we have made any solution like potassium dihydrogen phosphate or sodium disodium carbonate, or any potassium dihydrogen phosphate, any phosphate solution we have made, or any buffer solution made, and we'll add some OPA or TFA or some other some solutions to make a pH constant. So that buffer solutions, that solution which resists in its pH on dilution, so on dilution it resists the pH change in its value. Dilution in the sense when it goes into the column or when it mixes with a sample. So in that sense we need to say that this is a buffer solution which resists the change in pH, in simple language, right? So acidic buffer, mixture of equimolar quantities of weak acid or salt of this weak acid with a strong base is known as acidic buffer. We know, right? It is easy to understand. Mixture of ammonium carbonate, right? What is CH3COH and CH3CONA. right? Basic buffer, mixture of uh, equimolar quantities of weak base and salt of this weak base with a strong acid. So whenever we are mixing a weak base with a strong acid is known as a basic buffer, right? Like ammonium hydroxide, ammonium chloride are the example of basic buffer where we have basic basic base associated with it, like ammonium hydroxide. So hydroxide, right? Like ammonium chloride. So difference between HPLC and GC. This is very important and asked in almost all the interview questions. So you need to understand. Mobile phase, liquid solvent, inert gas, hydrogen, helium, nitrogen in GC. Sample type is non-volatile. We discussed this before also, right? In the GC, it should be volatile and thermally stable. And if it is not volatile, then how will analyze? But it should be thermally stable also at the same time. Operating temperature, usually room temperature to moderate temperature, right? And highly temperatures are required, like 200, 300, 400 temperatures are required to operate the gas to evaporate and to pass into the column. Separation principle based on interaction between sample, liquid, mobile phase and stationary phase. Based on volatility and interaction with stationary phase. We know this. Detector examples: UV, PDA, fluorescence, RI, right? Ultraviolet, photodiode array, fluorescence and refractive index detector. FID, fluorescence, FID, TCD, ECD and MS. Sorry, I'm forgetting the full form. So these are the different types of detectors used in the GC. Pharmaceuticals, proteins, peptides and antibiotics are the applications mostly for the HPLC, whereas in the GC solvents, acidyl solvents, hydrocarbons, volatile impurities. Right, in the sample state it is a liquid sample, it is vaporized sample. Cost of operation is moderate HPLC, and in the GC it is generally lower but requires carrier gases like helium, hydrogen, nitrogen. So that's why sometimes it may go up, depends on the cost of a gas due to some issues in the gas supply or something. So this is the basic difference between HPLC and GC. Based on applicability, it is used for separation of thermally stable. Simply someone may ask, based on applicability, what is the difference? So I have just listed this separately. It is used for separation of thermally stable or non-volatile compounds. It is used for separation of volatile, thermally stable compounds. In nature, 80% of the compounds are non-volatile, means used by HPLC. So we use HPLC more frequently than GC. Wherever in a company, if you go, there will be like 40, 50 HPLCs. There will be 25, 30, 40 HPLCs instrument, but there will be only 3 or 4 GC instruments in an industry. So that is the basic difference between both the HPLC and GC. What are the detectors used for GC analysis? Name at least 5. So the full form which I was mentioning here, the detectors I forgot, so you can mention here, you can read here. Flame ionization detector, FID, electron capture detector, ECD, thermal conductivity detector, TCD, photoionization detector, PID, flame photometric detector, FPD, and the nitrogen phosphor detector, NPD. So I've listed down 5. In case if you forgot any one, so you'll at least get to know about 5. So right, I listed down about 6. So that is all about my this session. And we do have also important videos like quality control with lab NLC system suitability test, HPLC GC, warehouse practice, 21 CFR. Please go and check out those videos as well. Thank you for listening.